In a previous tutorial, we learned about covalent bonds, the forces that create molecules by holding the atoms together within a molecule. While covalent bonds are strong and critical to life, there is a second category of bonds that is just as important. They are called intermolecular forces because they hold two or more molecules together. In other words, they work between molecules. Today, we will talk about the three main types of intermolecular forces: ionic bonds, hydrogen bonds, and van der Waals interactions. Then, we will talk about how these bonds apply in the study of biology. Intermolecular forces are often referred to as weak bonds because they have a fraction of the strength of covalent bonds. Weak bonds carry out the essential function of holding one molecule to another molecule. By the end of this tutorial, you should be able to not only identify the covalent bonds in this example of water molecules, but also the weak bonds that hold them together. Weak bonds are found everywhere inside the cell. They are the force that transforms phospholipids into a cell membrane, unites receptors and ligands together at the cell surface, and intertwines two strands of DNA into its classical double helix structure. In each intermolecular force, we will see the creation of a positive charge and a negative charge. Once created, these opposite charges are electrostatically attracted to each other. Hydrogen, ionic, and van der Waals bonds differ from each other by the strength and duration of their opposing charges. The first type of intermolecular force is ionic bonds. Keep in mind that while an ionic bond can occur between atoms to create compounds or salts, which we learned about in the last lesson, it can also occur between molecules to create many important biological interactions, which is why we include it in our list of intermolecular bonds. As a review, ions are created by ionization, in which an atom, or sometimes a small molecule, completely gives up an electron, forming a cation, or completely accepts an electron, forming an anion. Ions carry what is called a formal charge, meaning that it is a complete charge. The resulting charge is typically stable, meaning that it doesn't change. We have already seen examples of several types of ions, including sodium and chloride ions. While both are common in biology, some of the more common ions are formed from functional groups such as amino groups, carboxyl groups, and phosphate groups, which we will learn more about in the next unit. Ionic bonds always occur in complementary pairs. If one partner is positively charged, then the other partner must be negative. Notice how the charge of each of these partners spreads around the molecule like a bubble. This is because ions are non-directional. Attraction is equally felt in all directions and has no geometric constraints on it. Some of you may have learned in a previous chemistry class that ionic bonds are actually some of the strongest bonds that form, with a strength of 300 to 400 kilojoules per mole. This is true when the ionic compound is dry or in crystal form. However, in biology, ionic bonds are almost always dissolved in water, where they are susceptible to dissolution by water. The presence of water interferes with the ionic attraction and turns the ionic bonds into a weak bond. As we have said, the attraction between two oppositely charged ions is a weak force with an average strength of about 20 kilojoules per mole. Recall that in contrast, the weakest covalent bond measures about 100 kilojoules per mole. In order to better compare them with other weak bond types, let's summarize the essential characteristics of ions and the resulting ionic bonds. First, ions carry a formal charge. While some ions are atomic, many biological ions are formed from functional groups. Ionic bonds lack a specific direction. They also require complementary partners, meaning that a positive ion can only bond with a complementary anion and vice versa. Finally, we will compare their bond strength of 20 kilojoules per mole with the strength of other bond types. Now, let's review what we have learned about ionic bonds, their charge, and their strength. Ionization allows some atoms to achieve a complete octet in their outer shell, but leaves them with an unfavorable charge. Positively charged cations are attracted to negatively charged anions by the attraction of opposite charges, forming the intermolecular force known as ionic bonds. Ions have a complete charge that is the same around the entire atom or functional group. Ions are essential to several functional groups of biological structures, including carboxyl groups, amino groups, and phosphate groups. In a crystallized or dry form, ionic bonds can be even stronger than covalent bonds. In water, their electrostatic attraction is weakened by the tug of the surrounding polar molecules, making them the weak bonds, with an average strength of 20 kilojoules per mole. The second type of intermolecular force is known as a hydrogen bond. First, we will explain what they are, and then we will explore the nature of their partial charges and how they differ from ionic bonds. The naming of hydrogen bonds is unfortunate because students often wrongly associate the word hydrogen bond with a covalent bond between two hydrogen atoms, or a covalent bond between hydrogen and another atom. This is false. Hydrogen bonds are actually a weak bond that occurs between polar molecules. One of the polar molecules must have an electronegative atom like oxygen or nitrogen, and the other molecule must have a polarized hydrogen that is covalently bound to its electronegative atom. Hopefully, you'll remember from our last tutorial that the difference in electronegativity between hydrogen and oxygen, or nitrogen, creates a polar covalent bond which pulls the paired electrons farther away from the hydrogen. This pull of electrons is just strong enough to make the hydrogen partially positive, and the oxygen or nitrogen partially negative. The partial negative charge on the oxygen atom from one molecule can be attracted to the partial positive charge on a hydrogen from an adjacent molecule. This attraction creates a weak bond that we will refer to as a hydrogen bond. Hydrogen bonds, as you can see, are typically represented by a series of 3 dots between the associated atoms. Hydrogen bonds hold polar compounds together. This is a big deal because most of the molecules inside a cell are polar molecules, including water, which we will spend a whole tutorial discussing. Another prominent location of hydrogen bonds is the DNA double helix, where hydrogen bonding allows the A's, G's, C's, and T's from one strand to recognize and base pair with their complement on the opposite strand. There are a couple of features that make hydrogen bonds different from the ionic bonds discussed previously. First, the partial charges involved in hydrogen bonds are not complete ions. They are partial rather than formal. Second, they have different functions in the cell. And third, while ionic bonds are non-directional and non-specific, hydrogen bonds are different in that they are directional. This means that a partial positive hydrogen from a neighboring molecule can only associate at certain points defined by the location of the unpaired orbitals. In other words, a hydrogen bond won't form unless they're in the right geometric orientation. This directional charge is the reason water forms a regular lattice to make ice crystals. This crystal lattice actually pushes water molecules slightly apart, making ice less dense than its liquid form and giving ice the ability to actually float in water. The illustration also makes a very nice wallpaper, if you're a chemist or a biologist. Fourth, as with ionic bonds, hydrogen bonds involve interactions between opposite partners, like male and female. Since the terms positive and negative are already used with ionic bonds, in hydrogen bonds we identify the opposite partners by calling the molecule contributing the partial positive hydrogen the hydrogen donor, and the molecule contributing the partial negative charge the hydrogen acceptor. Fifth, hydrogen bonds have a range of binding energies that vary between 10 to 30 kilojoules per mole. The variance depends on the polarity of the covalent bonds from each molecule that created the partial charges. Now let's review what we've learned about hydrogen bonds. Hydrogen bonds only form between polar compounds, or molecules that contain a partial positive and a partial negative charge on opposite sides. Unlike ionic bonds, hydrogen bonds have a partial directional charge. Because hydrogen bonds are directional, they bond in a specific geometrical pattern that limits each molecule to predictably located binding sites. While ionic bonds occur between positive and negative partners, hydrogen bonds occur between hydrogen donors and hydrogen acceptors. Common examples of hydrogen bonding occur in water and in the DNA double helix. So far, we have presented methods for holding two or more ions together and for holding two or more polar molecules together. But what about nonpolar compounds? Is there a type of weak bond that might be used to hold two nonpolar compounds together? The third type of weak bond, referred to as a van der Waals interactions, does just that. In biology, van der Waals interactions occur between brief induced charges that appear and disappear when two nonpolar molecules are pushed next to each other. These instantaneous charges occur because the electrons in one polar molecule can start to influence or induce the electrons in another nearby bond. We will be using this squiggly line to signify van der Waals interactions in this video, but note that there's no standard chemical notation for this bond type. So the squiggly line only has a meaning in this video, where we hope it adds clarity. Van der Waals interactions play a significant role in several key biological structures. Cell membranes are one such structure. The cell membrane is composed of long hydrocarbon chains that form van der Waals interactions with one another. The fact that van der Waals interactions easily break and reform between any two hydrocarbon partners means that the membrane can be very mobile or fluid, while retaining its structural integrity. This fluidity could not be achieved using hydrogen or ionic bonds because they require specific partners. The folding of proteins is another fundamental process that depends upon the van der Waals interactions formed by nonpolar amino acids. By way of explanation, let's consider two hydrocarbon chains composed of just carbon and hydrogen bonds. Each of the bonds are nonpolar, so no formal or partial charges exist in either molecule that might hold them together. When hydrocarbon chains are placed in water, the water molecules are unable to bind with them. As a solution to promote bonding stability, the water molecules push the chains together in a process called hydrophobic collapse. This is why oil, which is nonpolar, and vinegar, which is polar, separate so quickly when you mix them together to make salad dressing. Thanks to hydrophobic collapse, nonpolar molecules can be close enough to each other to bond. We can illustrate what happens in a van der Waals interaction if we zoom in on a pair of oil molecules. We will focus on one of the nonpolar carbon-hydrogen bonds. The electrons making up the hydrocarbon bond are equally shared but constantly in motion. When a nonpolar bond gets pushed near another nonpolar bond, the electrons from one bond push the electrons from the opposite bond, so they avoid clashing on the hydrogens at the same time. As a result, the electron pairs start to synchronize their movements. As they move, the electrons create split-second partial charges on the hydrogens. One of the hydrogens will have a partial positive charge because the electron pair is temporarily located on the carbon, while the other hydrogen has a partial negative charge because it is temporarily holding the other electron pair from the opposite bond. The van der Waals interaction is a small attraction that is created from this slight, temporary separation of charges. As the electrons travel, the charges on each switch, but the opposite attraction and the resulting van der Waals bond remains in place. The van der Waals bond is the weakest bond because the polarity is slight, and because the electrons can, and do, come out of synchronization from time to time. Of the bonds we have discussed, van der Waals interactions are the weakest. Each interaction has a bond strength of only 3 to 4 kilojoules per mole. This means that any single pair of attracted hydrogens would be easily broken. But when van der Waals interactions occur at multiple points along an entire hydrocarbon chain, it creates a significant holding power between the two molecules. So, although they may be individually weak, van der Waals interactions are collectively strong. If we were to compare van der Waals with our previous weak bonds, we would see that the charge for van der Waals bonds is both partial and instantaneous, meaning it changes from instant to instant. It is important in holding cell membranes together and allowing them to still be fluid. It is also important in protein folding. The charge itself is non-directional, and unlike ionic and hydrogen bonds, it does not require any opposite partner, since any nonpolar atom can be either positive or negative at any instant. The strength, as stated, is only 3 to 4 kilojoules per mole. Now, let's review what we have learned about van der Waals interactions. Van der Waals interactions occur between nonpolar molecules, including but not limited to hydrocarbon chains. Although nonpolar molecules are uncharged, they are still able to bond together by way of two processes. First, hydrophobic collapse, which pushes the chains together so that they can bond over a short distance. Second, induced dipoles are brief charge separations within the molecules caused by instantaneous gaps between electrons as they rotate. A van der Waals bond has a relatively low strength of 3 to 4 kilojoules per mole. However, because van der Waals interactions often bind long chains together, they become collectively strong. In this final section, we will look at the relevance of weak bonds in a biological setting. We will talk about energy, melting and boiling points, and proteins and ligands. In several class periods, we will discuss the energy and flow of energy in biological systems. The formation and breaking of weak bonds is an important part of this flow of energy. A good general rule to remember is that the formation of weak bonds releases energy into a system by making the molecules in the system more stable. Breaking weak bonds generally requires an input of energy. Usually, this energy comes in the form of heat. For example, a pot of water has trillions of hydrogen bonds holding the water molecules together into a liquid state. Heating the water on the stove causes the hydrogen bonds to break between water molecules and creates water vapor. Later on in the course, we will learn about enzymes. These are proteins in the body that facilitate chemical reactions. The energy released by the formation of weak bonds between the enzyme and the chemical reactants play an important and essential role in allowing the enzyme to catalyze these reactions. As previously stated, individual molecules are held together by one or more of the 3 weak bonds that we have presented in this lesson. Many things that we consider as solids, such as a cube of ice, a stick of butter, or a piece of steak, are held together by these weak interactions. We melt a stick of butter by adding heat to it, which disrupts the weak bonds holding it together. By zooming in, we can see that the energy breaks enough of the van der Waals interactions to create a more fluid state. Heating our ice cube also creates a liquid, in this case by breaking some of the hydrogen bonds that hold the individual water molecules together. While melting ice breaks some of the hydrogen bonds, boiling water breaks all of the hydrogen bonds holding the water molecules to each other, which is why the water begins to fly away as vapor. Another important role of weak bond formation is the interaction between proteins and their intended ligands. Proteins are a large and varied class of related molecules that act as molecular machines in the body. We have already mentioned enzymes as protein catalysts for chemical reactions. Other proteins serve as signal receptors on the cell surface to identify and communicate incoming cellular messages. Still other proteins function as motors within our muscle cells, allowing movement. To carry out its function, each protein has to be able to identify, bind, and then release specific target molecules called ligands. Enzymes have to recognize reactants, receptors have to bind chemical signals, and motor proteins have to bind the related muscle fibers. Each one of these associations depends on the formation and release of weak bonds. Now, let's review what we've learned about the relevance of weak bonds. Weak bonds release energy when they are formed, but they require energy to be broken. Melting ice involves breaking hydrogen bonds between water molecules. Melting butter involves breaking van der Waals bonds between butter components. Enzymes are proteins that catalyze chemical reactions. Enzymes use weak bonds to associate with their chemical substrates. Proteins like receptors, enzymes, and structural filaments use weak bonds to associate with each other. Two strands in a DNA helix are also held together by hydrogen bonds into a double helix. Cell membranes, which are nonpolar, are held together by van der Waals interactions between phospholipids and membrane proteins. This concludes our discussion of intermolecular forces. This video tutorial was prepared by the Biology Education and Animation Research Project at Brigham Young University, Idaho, within the Biology Department. Video production and narration were carried out by Dr. Stephen Christensen. Animation and illustration of the various slides was carried out by Miss Cecilia Lyman. All images and artwork in this tutorial were created at Brigham Young University, Idaho, and we maintain all licensing and rights to these images.